Hi guys, it's another lesson of biology with Miss Cherry. Yay! Today we're going to look at monoclonal antibodies. So to begin with, you're going to start by pausing this, spending 5 minutes answering these questions, thinking about what you learnt from the last lesson, from the last video, and I'll come back to you in a second. So pause now and carry on when you're ready. So what you should have said for what is an antibody? From last lesson, we know that antibodies are proteins and they are produced in response to pathogens. Now, specifically, we are talking about bacteria because we're focusing on the humoral immune response when we're talking about antibodies. Humoral response. Now, the type of cells that produce antibodies is the B cell, okay, specifically plasma cells. And when we're thinking about antibodies, we know that they're there to help either agglutinate or opsonize or neutralize a pathogen, those bacterial cells that have got the antigens on them that we recognize as non-self. Okay. Now, how do we get large numbers of antibodies in a short space of time? We're focusing on the secondary immune response. And in this, we see the graph that we looked at. A primary infection gives us a low and slow production of antibodies, but after a second infection we get a rapid spike of our antibody concentration, and that's because we've got our B memory cells that are floating in the blood and persisting. Okay, remember we said about the immunological memory. And what they can do is once we experience that pathogen for a second time, they can undergo clonal expansion. So we can get lots of the same B cells being produced really quickly through mitosis using those interleukins. And we can get a lot of clonal selection. So we can produce lots of plasma cells. And those are going to be the ones that are producing all of those antibodies in a really quick and much quicker secondary response. So if you're not too sure what monoclonal antibodies are, good, I'm glad, because that's what this lesson is about. And we're going to establish what we mean by monoclonal antibodies. We're then going to take a look at how they're used in things like pregnancy testing, cancer treatments, and the ELISA test, or the ELISA test, depending on how your teacher says it, for HIV. And then finally, we're going to have a look at the ethical issues surrounding the use of monoclonal antibodies. So why are people for or against their use? Now, to begin with, we need to think about what mono means. Okay, monocle, a monobrow. What does that prefix, that beginning bit of the word, mean? And monoclonal means one specific type of clone of antibodies. So all of these monoclonal antibodies that a cell produces are identical. An example of this is when we've got a plasma cell responding to a specific pathogen. It's only going to produce one type of antibody, okay? And that antibody is going to be a monoclonal antibody specific to that pathogen. Okay, and we're going to abbreviate through the rest of the PowerPoint as MAB, because I'm a lazy writer and can't be bothered to write it out each time. Now, definition-wise, monoclonal antibodies are antibodies that bind to a specific single ligand or antigen. And by single, we're talking about one focused target. We're not talking about the fact that this antibody can go and bind to whatever it fancies. We're specifically looking at it working on one target molecule. Now, the reason why we say monoclonal antibodies is a completely different topic to the antibodies produced by the B cells is that because these monoclonal antibodies are those that have been made in a lab. Okay, and the idea of these is that they're used therapeutically for treatment, for testing. Okay, and then we're going to look at their use in things like cancer treatments, which is quite a novel approach, or pregnancy testing, which is the now commonplace approach for, or a more commonplace method of using them. Now, before we get anywhere, you need to know how we make those monoclonal antibodies. So we're going to start by taking a mouse. So we're using a live organism and we're injecting it with something that we want to target. We're going to inject it with a desired antigen. Okay, and that's the protein that we're going to find on the pathogen that we want to treat or test for. So that's what we're treating or testing for. Okay, so if we're looking at HIV, for example, we might inject it with a HIV antigen that cells that are infected with HIV might present with. Now, this mouse is a live organism, it's a mammal. It's going to have a very similar biological response to humans. So what happens is after that antigen has been injected, it's going to stimulate antibody production of a specific type. Okay? Now, that's all well and good, but those antibodies are within a mouse at the moment. So what we then do is we take the B cells that are there to produce those antibodies and we remove them. We effectively take the spleen of that mouse out. And we take those B cells and we fuse it with a myeloma, a type of cancer cell. And make sure you're careful with the spelling here. It's M-Y-E, not M-E-Y or just M-Y. Okay, and a myeloma is a type of cancer cell. It's going to really quickly divide, so hopefully in this instance it's going to get us lots and lots of copies of those B cells. But when we stick those two cell types together, we form a hybridoma. And you can see here in the diagram, we've got our B cell with some antibodies, and then we've got our myeloma cell over here, the yellow. And once we've stuck those two cells together, we are forming a hybridoma. And finally, what we do is we're going to reproduce that hybridoma loads and loads of times because it's a cancer cell, rapid division. We're going to get lots and lots, millions and millions of antibodies being produced, which we then collect and purify. We don't need that myeloma cell, the hybridoma, attached to the antibodies. We just want the antibodies themselves. And that is a pure sample of MABs. Now I want you to pause and do your own research, do some own revision and think about why people might have an issue with the way that monoclonal antibodies are produced. Think about the ethics around it, think about investigative purposes, things like that, and come back when you're ready to discuss. Now the major argument around why monoclonal antibodies are unethical, especially in their production, is the fact that this poor mouse gets infected and then it has to have a little operation to remove its spleen before any of these monoclonal antibodies can be produced. So is it really ethically right that we are infecting a defenceless animal that hasn't chosen to have this procedure done on it and subject it to a subsequent operation just for the sake of human development? Now we're going to take a look at the use of monoclonal antibodies in pregnancy testing. And these pregnancy tests are using monoclonal antibodies specific to the hormone HCG or human chorionic gonadotrophin. For exam purposes, HCG is absolutely fine. Knowing the full name is just a little bit of extra detail. Okay, now, HCG, or human chorionic gonadotrophin, is a hormone that's produced in the early stages of pregnancy. So it's not produced throughout the whole pregnancy cycle, just at the very beginning, around day 6, when the egg, or the fertilised egg itself, starts to implant into the lining of the uterus. Okay, you can stop now, you can have a think about how the monoclonal antibodies are used, using what you know about pregnancy testing, using the diagram to help. But if you just want to get straight into it, carry on watching. Now, monoclonal antibodies in pregnancy testing are conjugated to coloured beads. And because these beads are coloured, we are able to see them. It gives us the visual results of our test really, really quickly. And as you can see from that diagram, we've got pregnancy test working in 3 main stages, and we're going to go through those now. Now, the pregnancy test starts with a woman urinating onto the test strip. And if she is pregnant, then that urine will contain some HCG. On this test strip, as you can see in the little diagram, we have got some monoclonal antibodies, the Y-shaped molecules, conjugated or attached to a coloured bead. Okay, and we'll just put a little note here that conjugated means attached to. Now, when we've got a woman who's pregnant and she urinates on this strip, that HCG is going to bind complementarily to that monoclonal antibody, and it's going to form a HCG, the hormone, antibody complex with the coloured bead attached to it. And it's really key to note that these monoclonal antibodies in the test strip, so the test strip MABs, are mobile. Means they're free to move up the test, and that's really important. That's what help gives us our positive or negative test, and helps us to see whether the test itself is actually working. So moving up the test strip, what we should see happening is both unbound and bound monoclonal antibodies or conjugated monoclonal antibodies moving up the test strip. The paper inside is very, very absorbent, so any urine with these mobile monoclonal antibodies are able to move up to the test window, okay, the first line that we will or won't see. And what we've got in the test window is we've got another monoclonal antibody that's visible but is immobile. Okay, so this immobilized monoclonal antibody is specific for the HCG antibody complex, and it's only going to bind to a HCG molecule if it is bound to that initial mobile monoclonal antibody. Now, as you can see in the difference between the reaction site and the orientation of the monoclonal antibodies at the test site, you might have noticed that the fluorescence or the coloured bead is now facing upwards. Now, that fluorescent or coloured bead is pointing upwards, and that gives us the visible line. And it's this line that tells you whether you are pregnant or not. Now, even if you're not pregnant, you want to know whether the test is actually working, to check that you haven't got a false positive or a false negative. And the urine is going to continue down the test site. And again, we've got a selection of different immobilized monoclonal antibodies. So different MABs. And these ones are specific to the initial mobilized monoclonal antibody just down here. So even if you aren't pregnant, these unbound monoclonal antibodies from the reaction site are going to travel all the way down the test strip to the control site and rotate again and put the fluorescence or the coloured bead at the top. And this in turn means that we're going to get a nice visible line that will show that the test is working. If we don't get that coloured line, that second line visible, it means that we're not sure whether the test is fully functional and the results shouldn't necessarily be trusted. There could be a rip in the paper, there could be something else, maybe it's been tampered with, that gives you the result that you've got. So now we're going to think about how monoclonal antibodies are used in cancer treatment. And I want you to think about what we need for pregnancy testing and how monoclonal antibodies are used then, and to suggest how you think they could be used for treating cancer. Now you can pause it here again, and you can make some notes, you can do some research, you can discuss with me on Teams if you fancy, and we'll carry on in a second. So there are 2 ways that we can use monoclonal antibodies to treat cancer. And the first way we can do this is by triggering the immune system. So the monoclonal antibodies are going to trigger the immune response to kill the cancer cells. Now this is quite a difficult concept to actually happen, because if I've got a normal body cell, that has antigens on the outside that are triangular shaped. Cells that are cancerous, that undergo mitosis too rapidly, are my body cells just a little bit broken. So the cancer cells are also going to have those very similar antigens. They might just have slightly different shaped ones as well. So it's quite difficult to target and trigger the immune response to focus on those cells. Instead, what we can use is those monoclonal antibodies to flag up, or opsonize, the word from last lesson. We can flag up those cancer cells for destruction by the immune system. We can try to increase the rate of perhaps hydrolytic enzyme activity. So maybe we're releasing those lysozymes into the extracellular space. Or if it's really focused, really targeted, we can start using those monoclonal antibodies to trigger T cells, and specifically T killer cells, to produce that perforin that we spoke about. If you want to type in that link into YouTube at the bottom, it should come up with a nice graphic from cancer research that shows you how the monoclonal antibodies actually work to do this. The alternative way that we can use monoclonal antibodies is a way to carry the cancer drugs. Now, what we can do is we can stick our chemotherapy or radioactive substances that are used to treat the cancer to the monoclonal antibodies and target those to the cancer cells. And this is a way of combining both the medication and the targeted delivery that is going to help treat cancer cells far more effectively than having a whole bodily widespread chemotherapy treatment plan. Now, the final way you need to know about using monoclonal antibodies is the ELISA test, or the ELISA test. I say ELISA, don't at me about it. And that stands for enzyme-linked immunosorbent assay. You don't need to worry about this bit. Again, it's just a little bit of extra detail in case you wanted it. Now, the ELISA test is there to measure and detect antibodies in a blood sample. So we're not looking for antigens. And the idea of this ELISA test is to recognise whether you've got antibodies for a particular condition. And you need to know about this test in terms of HIV. So that is to say that your body, if you are infected, will produce antibodies to fight against HIV. And those should be circulating in the blood. Now, if they're circulating in the blood, the ELISA test is going to be able to pick those up regardless of the concentration and let you know that you either are HIV positive or not. So to begin with the HIV test, the ELISA test for HIV, we're going to start with a glass plate. And we're going to use glass because it is inert and inactive, so unreactive. And what that means is that I can stick HIV antigens, so proteins from the HIV molecule. And we're going to have a look at that in a few lessons' time, that are on the outside. So if I've got my viral particle like this, there's going to be proteins on the outside of them that flag up and make HIV. Okay, now these are going to be attached to the plate. And what I do then is I apply a small sample of the patient's blood to the plate. If I've got HIV in my blood, so I'll just quickly draw that in now, I've got blood, I'm going to be hopefully producing some antibodies to help fight that HIV. And that's what we can see here in the diagram. Okay, so these blue antibodies, these HIV antibodies, are produced by my body. Okay, by body to fight HIV. Now, obviously, that's not necessarily going to work very well, but it's nice to know that your body is fighting them. And those HIV antibodies are going to bind to those HIV antigens that are stuck to the plate. And what we've got there is the long chains of the antibodies facing upwards. Remember, we're undergoing complementary binding. So those two, antibody and antigen, are very, very specifically tertiarily structured. Now, after we've done that, we're going to wash away all of the blood. Okay, we're just going to give it a little rinse over, and that is to remove any unattached antibodies. I don't really think it's going to be very helpful if I'm looking for results to have loads of random antibodies floated around. So we just give them a big old wash off and we get rid of those antibodies by flushing away all of the blood. And now all that we've got is those monoclonal antibodies from the blood, the ones that I've produced in response to the HIV that I've infected with, bound to the antigens. Now carrying on, I'm then going to add a second monoclonal antibody. So if I colour in my picture very quickly, just on this left example, I've got the antigen that is fixed to the plate. I've then got the monoclonal antibodies that I've made. So I'm just going to put anti-HIV MABs from body. And then I am going to put this third monoclonal antibody on that is specific to the HIV antibody. So MAB specific to HIV antibody. And you may notice that we've got this little E here. And that E means that it's conjugated to an enzyme. It's got an enzyme stuck to it. And this is going to be really important in the next step after we've washed the plate again. We've got to get rid of all of those unbound antibodies and enzymes so we don't get an incorrect result. Now that enzyme is active, it's functional, it's bound itself to the monoclonal antibodies now, just here. And we're going to add a substrate. Okay, the substrate is complementary or specific to the enzyme. You don't need to worry about what the substrate is or anything like that, but we wash that over the plate. And if our enzyme is present because it's bound to the anti-HIV MABs, then that enzyme is going to catalyse the formation of a coloured product from the colourless substrate. And this is what gives us our positive test. So if you've got no anti-HIV MABs in your blood, that means that your body hasn't had to fight HIV. And what that means is that the monoclonal antibody for the anti-HIV antibody can't bind. And that means that the substrate stays colourless. And that's our negative test. Okay, so you can spend some time now, you can pause this video. I want you to go back over the video a few times and make sure you are confident in explaining how the ELISA test or the ELISA test happens and is used to diagnose HIV. So pause now and come back when you're ready. Now we're going to go back to studying ethics again about monoclonal antibodies. There's lots and lots of examples in literature and stuff for the way that monoclonal antibodies has been used for scientific research and to some benefit actually. However, you still have to be able to consider the ethics. Now could you please spend some time now looking at your textbooks, using your iPads, doing some revision, some research to make a list of the pros and cons of using monoclonal antibodies. No longer making antibodies, that's what we discussed earlier in the lesson, but specifically about their use. Okay, you can put those in your lesson notes, you can upload them to our OneNote area or your OneNote area. You can put them in a collaboration page if you fancy. And I should be able to see in about 10 minutes some evidence on your notes pages of the work that you've done and the pros and the cons of using monoclonal antibodies. Now the final thing we're going to look at is an example exam question or two. I've got about 9 slides of them, including the answers. So have a look at this question. This is from a specimen paper around 2014-ish. This test only detects the presence of HIV antibodies. Give 2 reasons why it cannot be used to find out if a person has AIDS. So you're going to look at the question Going to look at the resource. Pause now, give yourself 3 minutes to answer it, and we'll come back then. Okay, question 2. The solution will remain yellow if a person is not infected with HIV. Explain why. And remember, explain means give reasons for. Okay, so give a reason for the fact that the solution will still stay yellow. Pause again, 3 minutes. Get started when you're ready. A mother who was infected with HIV gave birth to a baby. This baby tested positive using the test. This does not prove the baby is infected with HIV. Explain why. And again, give reasons for. Now with this, I want you to look carefully. Why else might a mother who has HIV give the baby HIV, maybe only temporarily? So you've got another 3 minutes, pause it again and come back in a second. And finally, a control well is set up every time this test is used. This is treated in exactly the same way as the test wells, except that blood plasma is replaced by a salt solution. Use the figure to suggest 2 purposes of the control well. I've got 3 minutes again. I need 2 reasons of why we use the control well. Think back to pregnancy testing, think about the ELISA test, look at the steps involved and see if you can answer them. Now the first question was looking at 2 reasons why the test isn't useful to see if a person has AIDS. And you should have recognised that there's a distinction between HIV and AIDS. Now that is to say that they're linked, and if you have HIV for too long, then it can lead to AIDS, but they are separate illnesses. So in order to see if a person has AIDS, you need to look for symptoms. That's the first mark. Okay? And for the second question, or for the second mark, you need to make sure that you've mentioned about looking for the number of helper T cells to see whether they've been destroyed. Now, you might not have got that second mark because we haven't done the HIV lesson yet, but if you come back to this lesson once we've done lesson 2.20 and have another go at this question, hopefully you should have been able to get that. Now, for the second question, why will the solution remain yellow if the person is not infected? And this was hopefully quite simple. The HIV antibody isn't present if the person doesn't have HIV, and therefore the second antibody or the second monoclonal antibody will not be able to bind, and therefore there won't be any enzyme to catalyse the formation of that coloured product that gives us that positive test. So the results will mean that the lack of antibody means that there's no enzyme, and therefore the colour change won't occur. Now again, this next question required a little bit more background knowledge, and you might have had it, but if you don't, then this is a learning opportunity. Why doesn't a positive test in the baby prove that the baby is infected with HIV? And this is a lot to do with fetal development and how the baby grows. So actually, when we're in the womb and just after birth, the child is going to receive antibodies from their mum. So the blood flow through the umbilical cord and things like that mean that the child's blood is made partially from the mother's blood. And if the mum has HIV antibodies, which she would do because she's positive, the child is also going to have those. And the solution, therefore, will always turn blue for the first few months of life. The answer does say up to 18 months. That's in brackets. It's not necessarily required for the mark, but it is good to have that knowledge. And finally, the last question, what are the 2 purposes of the control well? You can show that we've only got the enzyme or nothing else causing the colour change, because if we're putting a saline solution in there, then that's not going to have any antibodies in, and that means that there's going to be no enzymes to bind to that antibody in the first place, and that washing away all of the solution is effective. Okay, so we have to rinse the plate a couple of times, don't we, to see the test results nice and clearly. Now here we've got another exam question, and you won't have seen this malaria example anywhere else so far. Okay, so malaria is a disease caused by parasites belonging to the genus Plasmodium. 2 species that cause malaria are Plasmodium falciparum and Plasmodium vivax. A test strip that uses monoclonal antibodies can be used to determine whether a person is infected by Plasmodium. It can also be used to find which species of Plasmodium they are infected by. A sample of a person's blood is mixed with a solution containing the antibody A that binds to a protein found in both species of Plasmodium. This antibody has a coloured dye attached. A test strip is then put into the mixture, and the mixture moves up the test strip by capillary action to an absorbent pad. 3 other antibodies, B, C and D, are attached to the test strip, and the position of these antibodies and what they bind to is shown in figure 7. Okay, so we're using this picture, we're using the diagram, and a little bit more detail that comes from the questions. Now for this first question, explain why antibody A attaches only to the protein found in species of Plasmodium. Explain why antibody A attaches only to the protein found in species of Plasmodium. So pause now, you've got 3 minutes, and play again when you're ready. Antibody B is important if this test shows a person is not infected with Plasmodium. Explain why antibody B is important. And you're going to want to think very carefully about pregnancy testing here. Notice that it's still a strip of absorbent material very similar to that absorbent material in the test strip of a pregnancy test. So pause again, 3 minutes, and go on when you're ready. One of these test strips was used to test a sample from a person thought to be infected with Plasmodium. Figure 8 shows the result. What can you conclude from this result, and explain how you reached your conclusion? So what do those results show? What are they infected with, and how do you know? Pause again. This time you've got 6 minutes, and we're going to go on to the answers on the next slide. So the questions and their answers are as follows. Why does the antibody A only bind to Plasmodium proteins? And what you should have got from this is that it's a very structural question, in so much as that you needed to say that the antibody has a tertiary structure. And this is complementary to the binding site on the protein. So the antibody A is complementary to a binding site on the Plasmodium proteins. Now for the next question, why is antibody B important? This was very similar to the second line on a pregnancy test. So it prevents false negatives, and since it shows that antibody A has moved up the test strip and it hasn't bound to any Plasmodium proteins, it's only specific to that antibody A from the blood sample solution. And finally, what do the results show? Well, you should have seen that the person is infected with Plasmodium, or it has malaria, and is specifically they're infected with Plasmodium vivax. That's because we can see the coloured dye where antibody C is present, and that only binds to the protein from vivax, or there's no reaction with the antibody for falciparum. I hope you found this useful. If you've got any questions, let me know. Post them on our OneNote. You can send me an email, things like that. And I will see you next time. Bye.